The meeting theme for today is Scientific Platform Success Stories. And I'm going to invite François Jean, our CNSP Western Canadian representative and Associate Professor in Microbiology and Immunology at the University of British Columbia. He's also the founder and former scientific director of the Facility for Infectious Disease and Epidemic Research, or FINDER, at the University of British Columbia. And he's going to introduce our first success story today.
Okay, thank you, Claire. Bonjour à tous. It is really with great pleasure and excitement that I introduce our keynote speaker today, Dr. Pieter Cullis. Pieter is the Scientific Director and CEO of Nanomedicine's Innovation Network. He's also professor in the Department of Biochemistry and Molecular Biology at the University of British Columbia. Pieter and his co-workers have been responsible for fundamental advances in the design and development of nanomedicines, employing lipid nanoparticle technology for cancer therapies, gene therapies, and more recently, the development of mRNA vaccine delivery for COVID-19. Pieter was elected a Fellow of the Royal Society of Canada in 2004 and was also awarded the Prix Galien, Canada's premier prize for achievements in pharmaceutical research and development in 2011. Today, Pieter will be highlighting the development of the LNP and their contributions for developing novel therapeutics and vaccine, including the new COVID-19 vaccine developed by Pfizer and BioNTech. Since Pieter could not join us today, his talk was pre-recorded on June 2nd. Following his presentation, which should be about 30 minutes, we will share with you a discussion, which should be about 15 minutes, with Pieter underlining his exciting and very productive journey in the development of LNP technology. Pieter will also share with us his vision regarding the next frontiers in this very rapidly evolving research area, and will also underline the importance of scientific platforms to achieve this important research breakthrough in Canada. Thank you.
Okay, thanks, François, and it's a pleasure to present to the CNSP. What I'm going to talk about, formal title is Scientific Platforms in the Development of an mRNA Vaccine Delivery System, but really an alternative title, and one that I'll use throughout, is a 25-year journey to enable gene therapies, which led in part, or to some extent, to the Pfizer-BioNTech COVID-19 vaccine. I have a couple of conflicts of interest. I'm a founder that are germane to this presentation. I'm a founder of Precision Nanosystems, and I'm founder and chairman of Acuitas Therapeutics. Okay, just to get down to some basics. What is gene therapy? And according to Wikipedia, it's a very general definition. Gene therapy is the therapeutic delivery of nucleic acid into a patient's cells as a drug to treat disease. The operative word there is delivery, and so that's the platform technology really that I'll be talking about throughout, which is a delivery technology to deliver nucleic acid polymers into a patient's cells. And so this is required for very basic reasons. The unmodified nucleic acid polymers are very rapidly degraded in any biological fluid. If you inject them systemically, they don't preferentially accumulate where you want them to go. They may not get there at all. And even if they get there, they can't penetrate into target cells because they can't get across the cell membrane. Now, this is an area of really extreme interest, gene therapy, because potentially any disease can be treated using this approach. And that's resulted since about 1990 in 2,500 now, I think, gene therapy trials. Most have used viral delivery systems. However, out of all of that work, it's an enormous amount of work and enormous expense. There's only been 6 drugs approved so far. 3 of those have used viral vectors, adeno-associated virus, and so those are the 3 indicated: Chimera, Luxturna, and Zolgensma. One of those is ex vivo chimera for CAR T-cell therapy. But now there's 3 others that have arrived on the scene quite recently, Onpattro and 2 COVID-19 vaccines. And I'll be describing the Onpattro and Pfizer BioNTech vaccine throughout this talk. So it's a situation that's changing. And why is it changing? The viral vectors, there's been a tremendous amount of really amazing work trying to get viral vectors to be more suited to gene therapy applications, but they suffer from a lot of issues. Limited genetic capacity. They're quite difficult to engineer and manufacture. They'll stimulate an immune response, which means that you can only give 1 dose. There's a variety of other issues, very expensive, toxic, can give rise to mutagenesis. And so it's been a fraught journey for viral vectors as gene therapy platform technologies. Non-viral systems potentially have none of those issues, but historically, they've had limited transfection potency and can be quite toxic in vivo. And so these issues are now being overcome using lipid nanoparticle delivery systems. And I'll go through a little bit in this talk as to how that has happened. So lipid nanoparticles that enable gene therapy. I'll describe quite briefly the design of these systems, and then go on to the Onpattro story. This is a drug to treat a condition known as hereditary amyloid transthyretin-induced amyloidosis. And just the program and the disease, and then the clinical trial results. And then I'll close with a few slides on the BNT162b2 story. This is the Pfizer-BioNTech COVID-19 vaccine. Now, going back to 25 years ago, this was the challenge that we took on, essentially to develop a delivery system, a lipid-based delivery system that takes nucleic acid-based drugs to the liver and enables by an intravenous injection, taking these polymers to the liver, getting out of the vasculature, and then being taken up by the cells, and of course then releasing the contents into the cytoplasm. So this is quite a ambitious objective, and of course your immune system is exquisitely tuned to try to make sure that this never happens in terms of getting genetic information into cells in your body. So just to start off in a very basic way, if you want to encapsulate a nucleic acid polymer, indicated by the double helix here, in a lipid system, then the simplest way to do this efficiently is to have some positively charged lipids there that will associate with a negative charge, and you can see intuitively how you'll end up with a hydrophobic entity that conceptually can be placed into a lipid-based system. Now, there's no cationic lipids in nature. They're extremely toxic molecules in biological systems. There only are net neutral lipids or negatively charged lipids. And the problem that these positively charged lipids have is that they'll combine with negatively charged lipids and essentially rip membranes apart. And so we were faced with the issue of how do we get an encapsulation using positively charged lipids without using lipids that are going to be so toxic as permanently positively charged cationic lipids. And so what we did was we tried a lipid called DODAP, which we had in the lab for some basic science reasons. This is an ionizable cationic lipid, and that just means that when at pH 7, or 7.4 physiological pH, it's net neutral. However, the primary amine gets protonated as you go down, say, pH 4. The pKa here is about 6.5 or so, and becomes positively charged. And so you have a system that at pH 4, about the acidity of a lemon, is positively charged, but is not charged at physiological pH, and so therefore is much less toxic. Now, what we found is we could formulate nucleic acid polymers into the lipid nanoparticles using a rapid mixing procedure, essentially where we dissolved all of the lipids, and this is just, there's 4 lipid components in addition to the ionizable lipid that's indicated in green here. There's a PEG lipid, polyethylene glycol lipid, and a structural lipid, phosphatidylcholine, and cholesterol. So we mix those together with water at pH 4, buffered at pH 4, with the oligo that we want to encapsulate in that medium. Now, very rapid mixing, one of the first things to fall out of solution is going to be this hydrophobic particle here, where you have the cationic lipid, the ionizable lipid, that's positively charged, interacting with the negative charge on the oligonucleotide. If you do this very slowly, of course, they're going to aggregate and you get a great big glob of material. But if you do it fast enough, you can trap them in limit size systems. And essentially, the lipids such as the PEG lipid will fall out of solution. These are surface lipids, and really stabilize the whole system in these limit size particles. Then we dialyze away the ethanol and raise the pH. And so this resulted in a class of lipids. This is indicated here by cryo-TEM, but this bar here is 100 nanometers, so they're about 60, 70 nanometers in diameter. They have a hydrophobic core. This is a cartoon form here that contains the oligonucleotide RNA, SIR, small interfering RNA or messenger RNA, whatever you might want. And so they're ideally suited to encapsulation of macromolecules, negatively charged macromolecules. We can get encapsulation efficiencies approaching 100%, stable, monodisperse, relatively non-toxic, as I mentioned, and highly reproducible. So this was the formulation side of it. Now we move on to the application for this disorder that I mentioned, transthyretin-induced amyloidosis. So this was our objective from about 2005 to 2012, is to develop these systems using siRNA to silence genes in the liver following an IV injection. Obviously, there's a lot of disorders one can target using a if you can silence or express or edit genes in the liver. So can these systems that contain the ionizable lipids, such as this ionizable lipid DODAP, can they silence genes in hepatocytes? And so they're quite simple systems. There's no targeting information on the outside. And as we discussed, the oligonucleotide is encapsulated in the hydrophobic core. Now, we assess the in vivo potency of these systems using a factor 7 model. Factor 7 is made in hepatocytes, it's secreted into the circulation. It's one of the clotting proteins. And so what we did here was to inject lipid nanoparticles containing siRNA to silence factor 7. So this gave a relatively straightforward assay whereby we could inject the lipid nanoparticles, and then 24 hours later assay for factor 7 in the blood. And so the extent to which the levels of factor 7 were knocked down would reflect the potency of the system. So this was a reasonably high throughput model. We could run 2 experiments a week and to go through a number of formulations, a large number of formulations to develop the best or the optimized systems. This just indicates the range, the types of ionized. What we found, to our surprise actually, was that the potency of these systems was extraordinarily sensitive to the species of the ionizable cationic lipid that we employed. We synthesized over 300 different species in this collaboration. This collaboration was done with a company in Boston called Alnylam. And we moved through to these to MC3, which has ended up as being the most potent. But you'll note really that these are not very different molecules. I mean, they're quite simple, quite similar. And the only difference primarily is in their pKa. But this just shows you the remarkable improvement in potency that we're able to achieve over this 5-year time frame. Moving from a dose of 10 milligrams of siRNA per kilogram body weight to get 50% gene silencing. That's what the red line is here. I cut off the y-axis there. But anyway, to move from the dose level of 10 mg per kg down to 5 micrograms of siRNA. So, you know, over 1,000-fold improvement, 3 orders of magnitude improvement in potency. Without increasing toxicity. So it was we ended up with a therapeutic index of over 1,000. So in other words, you give 1,000 times higher dose before we saw toxicity. So the results of this program was that we could silence the factor 7 gene using these lipid nanoparticles containing siRNA to silence factor 7. And by extension, we can silence any gene we want in hepatocytes. And so it's just a matter of putting in a different siRNA to silence that will silence a particular target protein. So the clinicians at Alnylam chose to develop a system, lipid nanoparticle system, to treat this transthyretin-induced amyloidosis. This is a disorder that affects about 50,000 people worldwide. It might be more actually, but these are people who are actually diagnosed with the disorder. Transthyretin is a protein that's synthesized in the liver, secreted into the circulation as a retinol binding protein. But if there is mutations in the transthyretin gene, this can lead to the formation of fibrils that will circulate in the circulation and deposit presumably in all tissues. But it has really nasty ramifications in the nervous tissue and in cardiac tissue. And so there's really no effective therapy for this disorder. It's really usually fatal within 5 years of diagnosis. It's a really nasty multisystemic disorder. This is just indicating CNS problems, gastrointestinal problems, kidney problems, neuropathies, cardiotoxicity. The neuropathies include difficulty walking, etc. Actually, that's usually one of the first signs you're coming down with this disorder, and it's rather a nasty indication because, of course, you'll have seen relatives. This is a hereditary disease, and you've probably seen relatives come down with this disorder, and the progression can be horrific, as is indicated here, over the 5 years of the course of the disorder. Now, one of the things about gene therapies is that conceptually, at least, you can develop therapies that are really very targeted to the disorder. And so in this case, seeing that transthyretin is causing the disorder, well, let's silence it in the liver. And so the hypothesis here is to deliver siRNA to silence transthyretin production in the liver, reduce the levels of circulating tetramers. The organ deposition of the fibrils, of course, will be decreased. Perhaps you'll get clearance, maybe you'll get stabilization and recovery. The main point here is it's a potentially simple solution to this devastating disorder. So there's a clinical development program started in 2012. It was phase 1 was completed on healthy volunteers, and then there was an 18-month study that was concluded in August 2017 that investigated the efficacy in patients. And so this is just the results of the phase 1 study, which established the dose and interval levels of the lipid nanoparticle siRNA to silence transthyretin, the levels that should be given. And what you note here is that at levels above about 1.15 mg per kg, you're seeing this very dramatic silencing or decrease in the levels of transthyretin in the circulation, which extend over 20, 30 days. And so on the basis of this, the clinicians chose a dose of 0.3 mg per kg every 3 weeks for the subsequent clinical trials. This now went on to the phase 3 study in about 225 patients. There was a treatment arm and a control arm, placebo. They got sterile saline, And the endpoints that were looked at here were the change in the neural impairment score at that 18 months, quality of life, ability, weakness, ability to walk, body mass, and so on. So these are standard indications for particularly for neuropathic problems. Now, the results were announced in September 2017, and the drug in this clinical trial hit the primary endpoint and all secondary endpoints. So it was quite I think this is certainly the best result of a clinical trial that I've ever seen. So in terms of the neural impairment score, it actually improved compared to the initial condition, and so you're not just stabilizing the disorder, but you're actually making people better. P-value here, p-value. I don't know why they had to bother to calculate a p-value. There's no doubt that this drug works, and similarly, that's true for quality of life. As for self-reported quality of life, muscle strength, gait speed, nutritional status, and so on. So this Onpattro, or patisiran as it was called during development, is a stabilizing and potentially curative therapy for a previous fatal disease. This was approved. This drug was approved by the FDA in 2018 for treatment of transthyretin-induced amyloidosis. It's the first FDA approval of an RNAi-based gene therapy drug, so it's a pretty big deal. So it dramatically demonstrates the power of gene therapies. So now I'm just going to go on to the last part here and so give you a few slides, or show a few slides on the development of the COVID-19 vaccine. And here we're dealing with messenger RNA as opposed to siRNA. So this is a much larger molecule. So the question here is, does the lipid nanoparticle technology enable mRNA-based therapeutics? And so it's exactly the same process that we envisaged for the siRNA, intravenous injection. If we can silence genes in hepatocytes, perhaps we can express genes in hepatocytes. So we're facilitating the intracellular delivery and then the delivery to the cytoplasm after being taken up by endocytosis. Now, these are messenger RNA. They're much larger entities, as indicated here. I've tried to do this to scale, but it's not quite. If I really did it, you would be able to see the siRNA here, which is a much smaller molecule compared to the mRNA. Now, it turns out that the ethanol dilution and rapid mixing process allows very efficient encapsulation. And this also results in delivery of mRNA that distributes proteins in hepatocytes. So this is the indication here for the encapsulation. And as you'll see, they look very similar to the systems by cryo-TEM that we had for encapsulation of siRNA. So this is, again, a 100 nanometer bar. The dosing these animals intravenously and then looking at expression in the liver, you'll see here, this is for luciferase. And you can see this is live imaging at 4 hours post-dosing, and you can see a dose-dependent expression of luciferase in these mice. Now, this was work done by Acuitas, a company I co-founded in 2009. And what they've done is gone on to develop improved lipid nanoparticle systems that are now, well, it's more than 30-fold now, get higher levels of gene expression in the liver for these mRNA systems using luciferase as a reasonably high throughput system. Now, this is where serendipity comes in. We were approached in about 2014-15 by Drew Weissman, who said, well, I need a delivery system to get messenger RNA coding for viral proteins into muscle and immune cells. Drew is an immunologist. He's been working for many years with Katerine Carrico to explore the potential of mRNA therapeutics as vaccines. And they'd found modifications of mRNA that really improved gene expression, but they still needed the delivery system to enable this to be used in vivo. And so, of course, this is a introduction of the of mRNA that's coding for a viral protein, and which then results in, say, muscle cells in presentation of those antigens on MHC class 1, or if they get into antigen-presenting cells, MHC class 2, and giving rise, Drew hoped, to a vigorous immune response. Some of the first work was done on Zika virus, and so this was published in 2017. So here, the mRNA that was encapsulated was the pre-membrane and envelope glycoprotein of the Zika virus. And the results were quite spectacular. That's total protection against Zika virus. And so injecting this mRNA intradermally in this case, and then challenging at 2 weeks or 20 weeks. And this is basically total protection against the Zika virus infection in these animals, just from one injection of the nanoparticle containing the mRNA. Now, in January of 2020, Cullis was working with a German company called CureVac, and on a rabies vaccine. And so in this case, there's an mRNA coding for the glycoprotein on rabies. And this is an actual clinical results. So CureVac announced positive results at a very low dose of 1 microgram of mRNA in this phase 1 study. So high levels of neutralizing antibodies that looked extraordinarily effective. And so at that point, Cullis was also working with BioNTech, and BioNTech was working with Pfizer on a flu vaccine. And so in January of last year, all efforts switched to developing a vaccine for COVID-19, given that the pandemic was emerging. So here, the messenger RNA is coding for the SARS-CoV-2 spike glycoprotein. And all the rest of this is history, which, of course, you're probably well aware of. This was announced in November of 2020, 95%, 94%, or it is 95% effective against COVID-19 in a great variety, across all cohorts, gender, race, age, etc. Well tolerated, and it's that Pfizer BioNTech is now manufacturing here. They set up to 1.3 billion doses. I think they're saying they're going to manufacture 3 billion doses this year. It was approved for emergency use in a variety of jurisdictions, including Canada. And so this is just real world evidence that this this vaccine works extraordinarily well for Israel, a country of 9 million people. Just showing that when it's to the point where these are fully vaccinated with two doses, but you can see the impact on the infection rate in Israel, and they're now back to normal. So it's been quite a journey from developing a system for delivery of nucleic acid polymers to the liver, to the first drug that was approved for RNAi-based therapeutics, and then to the vaccines, which were obviously developed at a very high speed. Vaccine applications are really just the tip of the iceberg. We're going to see vaccines against HIV, Zika I mentioned, perhaps a universal flu vaccine, malaria, but there's many other applications where we might need a protein being expressed that can be expressed using this mRNA approach, or edited for that matter. And then of course, there's the rare diseases that are again attackable using this type of technology. Just one point is that the process here is very rapid. Once we identify the protein that we want to silence or express or edit, we can produce the sRNA or the mRNA in a time on the order of a month or two. Encapsulation is very fast, a day or so, and have a new medicine that's available in a time on the order of a few weeks. And so this dramatically shortens the time frame for development of new highly targeted therapeutics. Just acknowledgements. This is the work of literally hundreds, probably closer to 1,000 people, but I want to particularly acknowledge Mick Hope, Tom Madden. I've been working with them for 40 years now, and one of the reasons that we've been successful is that we've held the team together over that long time period. And that also goes for people like Steve Ansel, Ying Tam, Barb Wee, Paul Lin, Sean Semple. I've worked with most of them for more than 20 years. And then there's a variety of people that are involved on the microfluidics side of things, which I didn't mention: James Taylor, Ewan Ramsey, Carl Hansen. People are Abutis, James Hayes, and Ian McLaughlin. Al Nyland, great collaboration with them. Marco Ciffolini in the chemistry department. These things work in the brain as well. My own group, and Drew Weissman, who I mentioned at the University of Pennsylvania. And so with that, I'll close, and thank you for your attention.
Okay, so welcome back. Bonjour à tous. First, I would like again to thank Pieter for such an exciting talk. Next, we'll share with you a short discussion with Pieter where he underlined his vision regarding the next frontiers in this rapidly evolving research area, which is the application of LNP technology, as well as his insight into the next frontiers in basic research. You will also discuss the importance of scientific platforms to achieve research breakthrough in Canada, such as the development of mRNA vaccine delivery for COVID-19. I would like to point out that you're welcome to forward your comments and questions for Pieter. He actually said he will be pleased to get back to you personally on your questions and comments. So I will make sure that he received them. And I would like to also point out that you should stay with us as we hear from Mia Selavi immediately after the short interview. So I thank you again for your attention, and merci à tous, and I will see you in the next session. So first of all, thank you, Pieter, for your very exciting talk. And more importantly, I would like to thank you, you and your team and your collaborator for the development of such a robust and very important technology platform, not only for gene therapy, as you pointed out, but also for mRNA vaccine development. So thank you again. So first, I would like to discuss a little bit with you what do you think are the next frontiers and research areas for your technology? I think you listed some bullet point on potential application, but I would like to hear from you is how do you prioritize? So what do you think? Which one you think you should be investigating first?
Yeah, it's that's a good question. Because there are so many things that one could go after. I mean, obviously, there's a lot of disorders. I mean, if you can have a protein you can use the liver as a bioreactor, produce antibodies that might be therapeutic utility. You can replace proteins that aren't perhaps being made properly. But for me, the main thing right now is to get past the liver. Yeah. We're good at transfecting hepatocytes, but I think there's a push now to say, okay, well, can we transfect bone marrow? Can we transfect the lung? Can we get to the eye or the brain? And so those are the the areas in which we're focusing now. In order to do that, one has to get to develop delivery systems that circulate for much longer in the body. The systems we currently have are quite well targeted to the liver, actually, but it means that within 15-20 minutes after injection, 80-90% is in the liver. Mm-hmm. And so what we're doing now is developing systems that circulate for a longer time, and so therefore we will have access to tissues such as bone marrow, which we're starting to get quite good at. And also other tissues in the body. And I think the thing after that is going to be, well, how can you turn them on and off in one location versus another? You don't want to have a global expression of something that's maybe only needed in one tissue. And so the problems are going to continue for quite a while yet, and they're really quite fascinating ones.
Yeah. So I think from a molecular virology perspective, and keeping in mind the challenges for many years for vaccine development for HIV and HCV, I think this is certainly for those two specific viruses that can be actually transmitted or be present in the same individuals at the same time. That could be very powerful to develop such a technology. What I wonder though, have you thought of basically targeting multiple pathogens at the same time? So for example, by combining mRNA encoded by two different viruses, so example HIV, GP120, and perhaps E1, E2 of HCV. So multiple mRNA delivered through your technology. Is that feasible, do you think?
Well, it's very feasible, yes. People haven't really done it yet, but there's nothing to say you can't do that. Now, you do it two ways. Of course, you could just make two separate formulations and then mix them together, or you could have everything there starting from scratch. But yes, it should work, and there's intrinsically no reason why it wouldn't. As I say, right, we're in one of those stages where the gates have kind of opened, and there's just a lot of things to do, and so it's certainly created enormous excitement in the field. The amount of effort going on in lipid nanoparticles now is probably 100 times more than was in place about 2 or 3 years ago.
Yeah, another I think important area is I think you alluded to this specific one is therapeutics. So I think mRNA-based delivery is great, but obviously based on the work you've done with RNAi and even mimic of microRNAs or anti-microRNAs, one would think that this would be a very powerful approach or a strategy for developing again antiviral treatment. So what this antiviral treatment strategy is compared to the mRNA-based vaccine, is in terms of thinking about priority moving forward, is that high on the list, or this is like in the middle of the list?
Yeah, the short answer is that, well, I'm not an immunologist, but one of the, I'll give you an example of the kind of things that can be done, which actually are still mRNA based. You can make mRNA that say codes for the heavy and light chain of an antibody. And some of the work that Cullis has done with Drew Weissman is to say, okay, well, let's make an antibody against HIV, and then challenge the mice with HIV, and again, you get total protection. You get the antibody being expressed for 7 days or longer, and no protection against HIV. Whether that's going to be good as a therapeutic or not, I don't know. But certainly as a preventative for high-risk people in high-risk situations, you can see that being you have utility for say subsequent pandemics or whatever. There's a large number of different ways that you can deploy this approach. That's for sure.
You're going to be very busy, it seems to me. I think that this is there's so many applications I can see that are actually extremely important to work on. The last question on this section is biomanufacturing. Is that, I mean, is how challenging it is for your technology platform. Is like I can imagine you probably need kilograms of those liposomes. So where do you reach out? Do you get support here in Canada, or do you need to reach out to other countries to scale this up?
Yeah, this is really a multinational scale-up process. The lipid nanoparticle formulation was licensed by Cullis to BioNTech, and then in turn, of course, when they through the Pfizer. So when they're scaling this up, one of the advantages, of course, is that the dose levels. I mean, it's only 30 micrograms of mRNA. Now, the amount of lipid that's involved is about 20 times more by weight. And that's, it's cholesterol, it's the ionizable lipid, a bit of PEG lipid, and a bit of phosphatidylcholine. So most of those, well, some of those constituents are quite easy to make. It's the ionizable lipid and the cholesterol that tend to be the bottleneck. So that's one of the, from a straight procurement point of view. From the manufacturing point of view, that's not really a bottleneck at all. It's turned out to be it's really quite straightforward, because you're really just mixing the lipid dissolved in ethanol with the mRNA dissolved in a pH 4 buffer. And then subsequent, so there's just a mixing process, and then you dialyze away the ethanol and raise the pH to physiological pH. So that's not, the manufacturer has not proved the issue. The mRNA has not proved the issue, to my surprise, actually. I thought mRNA would be a limiting factor. But the one that is causing a bigger problem is the cholesterol. The FDA prefers that you use fully synthetic cholesterol, and it's the synthesis of cholesterol is not a simple matter. Of course, it's highly available in nature. We can get 100 milligrams or whatever out of an egg yolk, but the fully synthetic varieties are more difficult to obtain. So that has proved a bit of a bottleneck.
Okay, thanks, Peter. So now I've got a few questions related to the importance of scientific platforms in the success in developing such a technology. So could you comment for you how important the I guess the support and the progress I will say made in Canada over the last 20 years in terms of financially supporting development of scientific platforms. So give us your perspective on this through the 35, 40 years of what you have been involved into this. You probably could identify a turning point during that time in terms of the importance of that support to develop a technology. And do you think we're in the exponential phase now with moving forward, or do we plateau? So what's your perspective on this?
Well, first of all, I have to thank CIHR, before that MRC, for, I guess it's been over 40 years of continuous support. So that's really played a big role. I guess the other part is that I realized early on that if you're going to get something into the clinic, then at some point trying to do it through the university lab doesn't work. And I mean, the amounts of money are just too large. If you want to run a clinical trial, it's millions of dollars. And so I've been able to start up companies to be able to raise substantial amounts of money to develop the from, say, a bench scale where you're making lipid nanoparticles for one application or another, to the level at which will support a clinical program. And that, I think, is that transition that's really important, because something that works at the bench level, there's very often big problems in getting it to actually work at a industrial level. And these are not minor, people tend to think of scale up as being straightforward, but it's not. You have to have processes that are robust as you increase the scale of the operation. And we were lucky in a sense that we had the company involvements, which allowed us to really make that investment to, say, scale something up from making enough for a mouse model experiment to something that you could actually use in a clinical trial setting. So it's a very expensive process.
Yeah, no, certainly. So then a related question is, I guess, in terms of training of next generation of scientists in Canada. So again, your perspective on access to state-of-the-art scientific platform across Canada, across the different university, and those very important partnership with the industry. So are we going in the right direction? So give us your
Well, I've been encouraged on that a bit recently. I mean, I applied for a nanomedicines innovation network at NCE from the federal government, part of the NCE program, which was successful. And so we were funded in 2019 on that. And that's been a very good instrument actually for involving right across Canada. I think we have well over 40 investigators now. And so we're able to provide, state-of-the-art processes and platform systems that people can then go and use in their particular animal model, or whatever expertise that they bring to the network. So that's that kind of support, I think, is essential. Where you're really enable that collaboration between groups. I mean, we're quite good at formulation. Other people are good at, say, a particular animal model, and ranging from, some Alzheimer's model to a leukemia model, whatever it is. And then you can bring some, the combination, you can bring a reasonably sophisticated, say nanoparticle solution, and test it out in a sophisticated animal model that is well characterized. And so that's the kind of thing that these centers of excellence can support, and it's that's an absolutely vital part of the process.
Okay, so my last question, Peter, is it really about the lessons learned from the COVID-19 pandemic in terms of how important it is to support basic research and expecting the unexpected. I guess that's a way to put it. So I guess again, based on your very broad experience, not only in the academic but the industry. So what are your wish moving forward in terms of addressing and navigating our way through the COVID-19 and the variants of concern, and with other pandemic coming along? What would you like to say in general in terms of supporting research in Canada moving forward?
Well, yeah, I think this is something that is very important, I think, to transmit. That I don't think people quite realize that by supporting research in universities can have a dramatic impact on your personal health. And obviously the mRNA vaccine story is a very good example. And I think as scientists, we need to really get that word out there. I think some of the general population probably thinks, giving all these research grants to professors in universities, is that really something we're getting our money's worth for? And there's many ways that we can point out that that's true. But I think this is a particularly obvious one. The other part, of course, is that we were in the right place at the right time, but it was a very lucky circumstance in that sense. And when the results were announced last November, that was 95% protection against the infection by the SARS-CoV-2 virus. That was, to us, astonishing. It was it's very hard to predict something like that. And so it's a combination of good luck, but also the value of the basic science going on. That's in my lab's been going on for 40 years. It's proven to be pretty worthwhile. So.
Okay. Well, that's all I have for you, Peter. Again, thank you so very much for your great talk and being such a fantastic ambassador of research for all of us in Canada. And I know you're going to be very busy with the next 5 years or so with all of the application of your technology platform. Thank you so much again.
Okay, thanks, François. It's a pleasure.
No problem.